Hello students, today we will discuss development of dicot embryo. So before that, let's discuss what is embryo. Embryo means it is an early stage of development of a multicellular organism. It is an unborn or unhatched offspring in the process of development, or simply we can say embryo means zygote which undergoes several developmental stages and finally forms the embryo. So here this one is the zygote. Zygote is diploid. Here ploidy is 2n. It undergo mitotic division to produce two cells, basal cell and terminal cell. Basal cell is towards the micropylar end and terminal cell which is towards the chalazal end. These two cells undergo several developmental division and form the embryo. For our convenience, let's start with the basal cell. Basal cell, it divides transversively and forms 7 to 10 suspensor cell. This upper one is terminal cell, and here this dark green color represents the basal cell. Basal cell continuously divide and form 7 to 10 suspensor cell. The last suspensor cell, it enlarges, which is termed as hysteria. Hysteria provide nutrition to the embryo, whereas these small cells are called suspensor cell. The last cell of suspensor cell, which is towards the terminal cell, is called hypophysis. So, what is the function of suspensor cell? Suspensor cell simply push the developing embryo or the terminal cell deep into the endosperm, where it will get nutrition. Next is division of terminal cell. After division of basal cell up to 7 to 10 cell, last cell is enlarged and called hysteria, and the cell which is towards the terminal cell is called hypophysis, and these middle cells, neck-like cells, are called suspensor cell. Now divisions takes place in the terminal cell. First it divide into two cells. This is called two-celled embryo. Now further division takes place to form four-celled embryo. Like this, these terminal cells divide continuously and forms a proembryonal octane. Proembryonal octane means these four cells again divide and forms 8-celled stage. This 8-celled embryo, now it is called proembryonal octane. This proembryonal octane now undergo periclinal division. Periclinal divisions means such division in which a cell produces an outer cell and an inner cell. For example, this is a cell, it produces an outer cell and inner cell. Similarly, here also in the proembryonal octane, the cell which undergo periclinal division, it produces two cells, one outer cell, one inner cell. Like this, at the center 8 cells are there, and at the periphery also 8 cells are there. The peripheral cells, these are called dermatogens. Later on this dermatogen develops into epidermis. Here the largest part of suspensor cell is called hysteria, which provide nutrition, and this neck-like structure is called suspensor cell, and the last suspensor cell which is towards the terminal is called hypophysis. So let's discuss which part will transform into which part in the embryo. Here the four cell which is towards the hysteria it gives root cap and root tip, whereas this 4 cell which is towards the hypophysis, it gives the root cortex. Here in the terminal cell, the lower 4 cells will develop into hypocotyle, and the upper 4 cell, it gives plumule and cotyledon. These terminal cells towards the chalazal end, and here the suspensor cell as well, the hosteria is towards the micropylar end. So what is the function of suspensor cell? Suspensor cell simply push the developing embryo into the endosperm so that it will absorb the nutrition from the endosperm. Now this structure of embryo is called globular. So it is called globular embryo, as cells are divided and forms a globular structure. It is called globular embryo. Now division takes place continuously inside the ovule, where space is not enough. That's why after continuous division it forms a bulge structures. Two side bulge structures are produced, and at the center of these two bulge structure, another structure is there that gives rise to plumule. So now this embryo is called heart-shaped embryo, as the terminal cell divide continuously and bulged out at the two ends. It forms heart-shaped embryo. Now division continues, and here these two bulge structure continuously divide and forms a horseshoe shaped structure. These bulge portions develop into horseshoe shaped structure, and in between this structure it gives plumule. Other parts are the suspensor cell. It gives the hypocotyle, then the lower 4 cells that gives a radicle and root cap. So this is the dicot embryo, which is also called torpedo stage. Here we can clearly see the bulged portions develop into two cotyledon. In between the two cotyledon, this structure gives the plumule, and the lower part of the suspensor that gives hypocotyle. Now this structure gives the radicle and root cap. After development of the embryo, this hosteria cell degenerate. So this is the mature dicot embryo.